Dr. Harish sir for the invitation and opportunity for me to share some of the research work that I am and our group is doing. I just took one case study where we are working on a particular disease that is called febrile disease. Over a period of time, I will also, in the presentation, I will discuss why we are taking this particular disease, why we are working, what is the aim and other stuffs. Just to give you the information, kind of before I start with this presentation is majorly it is into academic sector. Okay, so I am into PS University, so into academics, and the reason in the presentation is one can publish a paper in a good impact factor using all the open source tools. Okay, so in the overall presentation, whatever I'll be covering today, I have not used any commercial tool. Okay, everything is open source, and we can still aim to publish in a good journal having like 3 impact factor. So the same work I am presenting here. So one of my student, Shravan, I think in 2018, we worked on this particular project. It's an 8th sem project, basically 8th semester BTech or BE biotechnology project where we worked on, and then we could do some more additional experiments after the finishing the project, and then we could publish. So the intention why I'm giving you this introduction is in the academic sector, even using all the open source, completely dry lab. Okay, so no wet lab work is yet done, but we are doing now for the validation. We have already started to check whatever the molecules that we identified is going to give the reasonable expected inhibition or not. So we are conducting that one, but still we have not received the wet lab report results. But yes, it is possible to do the completely dry lab work and use all the open source platforms, and high impact factor journal publication is quite possible to do. Okay, so how we can design the experiments and what are the difficulties that we will face over a period of time, and how to troubleshoot. If time permits, I can also show you the kind of a comments that we receive from the journal when we communicate, and how we can address few of the queries from them, and how we can publish. So that's the overall thing, assuming that most of you know with the docking. Okay, so just want to know if how many of you have performed the docking before. So you can just reply me back in the chat saying like done or docking. If you know the docking, you can say like know the docking, something, so that I will get to know like how many of you are aware of the docking. Okay, if you can just type done. So if you say like done, I assume that you have performed the docking earlier. Okay. Okay. Still, others can also reply back. Dynamic simulation also, if somebody has performed, you can also say like DS, dynamic simulation, DS done, so that, or what I can give an option is better to say like those who have not done, better to say like DS not done, because I think looking into this one, many have done it. So I just want to look for those who have not done it. DS not done. At least, is there anyone? Okay. Okay. So we have a mix of both the set of people who have performed the docking before. There are few set of people who have not done the docking before, and also with the dynamic simulation. Now, even to just to give you the brief introduction about what is docking, or what are the different types of docking that one can perform. So I have just put a screen showing like what are the different types of docking that one can aim for or one can perform. So this is a kind of a work that I'm working, where we want to see how a particular dye binds to a cellulose matrix. So whatever the fabric that is there, so how a dye binds to a cellulose, and this can be considered as a carbohydrate ligand docking. Okay, so it is possible to perform. We have the crystallographic coordinates of the cellulose. We can just download the cellulose crystallographic coordinates. Then we can generate its matrix using mercury, then save it as a PDB. Then we can load it as a macromolecule in the PYRX and perform the docking, just to get some hint how the interaction is. We can perform protein-protein interaction, that is here. So using this protein-protein interaction also, we will get to know the region. Okay, I have just put the stick representation here in the core if you look into. So we will get to know the region where the interaction is between A chain and the B chain. If we take as two proteins, how is the interaction? And we can also get to know how many hydrogen bonds and how many hydrophobic interactions, all those things. One can perform the DNA ligand interaction. This can be done using ClusPro server. So we can do the DNA ligand interaction. And when it comes to DNA interaction, we have two types of experiments needs to be done. One is how the ligand binds to a major groove or a minor groove, and also we need to check whether the ligand possesses intercalation, whether it can go between the DNA and like intercalator. So that also we can perform. This is a usual study that we do, protein ligand interaction. This is quite common. We can use Autodoc 4.2 or Autodoc Vina, which are freely available, and then we can perform the docking to see how is the ligand binding to a protein, and we can have number of conformers to check where and all the possibility for the ligand to interact. And this is also the same DNA ligand interaction, just like this, just in a different representation to show this is binding region. Then in any of these cases, okay, whether we do protein ligand, protein or carbohydrate ligand, DNA ligand, whatever, even we can perform the RNA protein docking. Okay, so a huge varieties of docking studies we can perform. We can generate the secondary structure of the RNA, and then we can check how the RNA can bind to a DNA if it requires, if the RNA is interacting with the protein, then that is possible. DNA protein docking, that is also possible way. So we can perform based on the things. And this is a 2D representation. Okay, in most of the papers you would have observed such representation. This is called lick plot representation. So how exactly a ligand binds to a protein, and all these are the amino acids. I have all these images in the next slides. So where exactly the hydrogen bonds, how many hydrogen bonds, to which amino acid the hydrogen bond formation is there, how many hydrophobic interactions are there. So since it is slightly difficult to interpret in the 3D at the first instance, so we always generate a 2D first, then understand the number of hydrogen bonds, the kind of a bond length, and then the hydrophobic interactions. That helps us to decide, do I need to generate the 3D next level. So if we have a good result, and if we have a promising result here, then we can convert this into a 3D and generate something like this or something like this or whatever. Then we can do it. So this is how the overall, based on the type of the target we have, we can perform the docking. In the present study, whatever I'm presenting, it is majorly with the protein ligand interaction, and also in one stage we performed the protein interaction. So both I will discuss. So why what exactly this pebrine disease refers to? And uh, pebrine disease is a microsporidian disease caused by Nosema bombaceae in Bombyx mori. And if you look into this image, A is the healthy one, healthy silkworm, and B is the pebrine infected silkworm. So for the silkworm, if it is infected, then the black spots we will observe, and then majorly this has affected the whole silk industry, even in the France in 1845. Okay, led to the fall of French and Italian silk industry. So even in the present situation also, this has a serious side effect in the silk industry. And if we have any one or two silkworms infected, so it is going to affect the whole batch. So in other words, we need to discard the entire batch just because few infected silkworms are there. So there is a high rate of horizontal transmission. If we just take an example, if 3% of the larvae in first instar stage are infected, then 50 to 60% will die by the time they reach the adult stages. So that is the level of infection it has. And then we will not have a good quality silk if it is infected. So it is going to affect the entire industry. This is one reason why we have taken this pebrine disease. And also at the academic sector, okay, it is relatively easy to work with these because we do not need to follow a very stringent guidelines to conduct these experiments. So it is feasible. Based on the feasibility, this is also a good opportunity where we can commercialize at a later point of time if we identify a drug molecule. So despite such high pathogenicity, there is no clear evidence or elucidation of the pathways. Not much of a work is done. Okay, really not much of a work is done. There is no drug actually available in the market for this, though it has affected the silk industry vast. Even now also it is affected, but there is no standard drug available to control this disease. And there are few papers where they said like there are many proteins. Okay, there are many proteins that we can use it as a target. We just started with the two proteins initially, I think in 2018, serine protease inhibitor 106. It is referred in short as SPN 106, which helps in the host immune evasion. That means this protein supports the pathogen to bind to the protein, bind to the host, and then do the evasion. Those things are there in the coming slides also, so that we can better understand on what basis I have selected this serine protease inhibitor as a target protein. And we have another protein called spurval protein 26. It is also observed to be responsible for the facilitation, the attachment of the pathogen. This also is there. There is one beautiful image by which we can better understand. So on what basis we can take these proteins as a targets. So another reason why we took this particular pebrine disease, the rationale, if we look into, is in our department in PS University, we have also developed a solution called a pebrine solution, or a PS pebrine PP solution, which is trademarked. And the beauty of this solution is we can remove off the artifacts and helps in the visualization. In other words, when we take a set of silkworms, and what is the setup that people follow now is, you will take the silkworms, look under the microscope, and if we find any infected silkworm under the microscope, if we observe, then if there is any infected, we will not consider the entire batch. The whole batch of silkworms will be removed off from the pipeline. And there are many instances where just because there are some dots, those are like false positives, so this was discarded. And there are few cases where it is too difficult to observe these spots also, that the infected spots, just as I showed here. So it was too difficult to identify these dark spots also under the microscope. So to avoid all these things, in other words, to increase the signal, so we have identified a solution. We have formulated basically a solution, and that solution, when we apply to a randomly selected silkworms, and when we check, it will increase the signal, okay, and decrease the noise. In the way, when we look under the microscope, it was very clear. It will highlight only the silkworms which are infected, and then we can take a decision that we can do whether to discard, or is it by chance there are some spots like that. So this solution is now has been allocated a trademark and is being marketed by PS University, and we are selling it out. We are selling it to most of the silk related organizations even in North India, and it has given a very good result. um, that means it made a life easy for identification of the spots and to discard, and also a commercialization angle for the university. Then what we thought is it possible not just to identify these spots or to identify these infected cocoons from the healthy ones? Is there a possibility that we can design a drug molecule, okay, so that we will not have this infected ones? Or is there a way by which we can spray to the mulberry leaves, or any different methods? We are not yet decided the mode of administration by which we can control, but is there a way by which we can make some attempt to control this disease? So in that attempt, to start off, we started with the bioinformatics approach, and then through bioinformatics approach, we did the literature to understand what exactly this disease is, how many proteins are involved, which protein we can target, and how is the mode of infection. All those things we studied, and how much information is available in the literature. Based on all those things, we have come up with the two proteins that we can work with, that is SPN 106 and SWP26. So what one should, whenever you are doing a research, so one should also think of is how feasible is to continue, not just as a publication. Okay, publication is just a byproduct of your research. So along with that research, especially with the dry lab work I am mentioning, is it possible that the identified molecule can be tested either at in-house within our campus or in collaboration with someone? How feasible it is to test, that is the first thing, and how much cost it is going to, how much cost it will be, and what is that new information that we can generate out of our research. That is also important. In a cases like this, Febrin, there are hardly very few groups that are working in the world. Okay, so not many groups are working. We do not have a crystal structure in the PDB. Nobody is solving these structures. So this particular work is a kind of a like ambitious project, people initially said. And even in this is in 2018, in 2019, okay, we took another protein. Even the reviewer from the journal himself commented like it's a very ambitious project, because we took some target some template which is far off, not closely related. So with all these possible ways, we are making an attempt to generate the data, okay, or better understand the disease. So because nobody has worked, just because nobody has worked, we cannot make a decision that we cannot do, because not sufficient data is available. Bioinformatics is all about generating the data or predicting based on the available information. So in that particular sense, okay, we took the proteins where we have minimal information and conducted the experiment. So one such target we took is SPN 106, so which belongs to a serpin family, responsible for the host immune system evasion, and it affects the melanization pathway of the Bombyx mori. Bombyx mori is nothing but the silkworm. Okay, that is the host. Nosema bombycis refers to the pathogen. So Bombyx mori is most important in inhibiting the pathogen propagation. SPN106 might interact with the beta glucan receptor protein to downregulate PPO. So it is there as a whole pathway here. So what happens is the melanization process happens, and in the typical melanization process, prophenol oxidase gets converted into phenol oxidase using this prophenyl activating enzymes. So this is the enzyme which activates, the conversion happens, then the melanization process happens. So the melanization process is typically the same like whatever our melanization process is. So it helps in increasing the immune system activity, but because of this inhibition of this Nosema bombycis, ENB is Nosema bombycis serine protease inhibitor, SPN. So when it inhibits, the melanization process is inhibited. Because the melanization process is inhibited, so the immune system becomes very weak. And because the immune system becomes very weak, so the pathogen enters and multiplies inside the silkworm, and the organism is going to die. So just to get you the same thing here, so Nosema bombycis, okay, this is the typical procedure. Beta glucans will reduce the beta glucan recognition proteins. This will generate the serine protease cascade, then the PPO cascade. Then this is the typical process. Then the melanization process is going to happen. But what is this Nosema bombycis pathogen? This is the pathogen. What it does is it will generate this protein, Nosema bombycis serine protease inhibitor 106. So this inhibitor blocks this cascade, and this cascade is blocked. So in other words, the melanization process is blocked. Because the melanization process is blocked, so the whole immunity of the organism is going to be very low, and over a period of time it will die. So we also need to look at whenever we take any proteins. Okay, so if we have some minimal information like this, so this is the only paper where we have a maximum amount of information for this disease. Okay, so we took this rather than experimenting with other proteins where we have a minimal information. This was quite a good amount of information, and to test with over a period of time if we want to test as a wet lab to check whether the drug is going to work or not. So we can design the experiments where the typical process of prophenyl oxidase to PO, if it is happening, if we induce this serine protease inhibitor, whether it is going to affect or not. If it is affecting, then is there a possibility that we can develop a drug that will inhibit this? So if we critically look into this pathway, we are designing an inhibitor to an inhibitor. Okay, this Nosema bombycis serine protease inhibitor, SPN, serine protease inhibitor 106. This serine protease inhibitor is a protein, and this is blocking this cascade. So we need to block this inhibitor so that it will not block this cascade. So we have to design an inhibitor to this inhibitor, so that this cascade melanization process will happen as a normal one. So of course it took some amount of time to understand this pathway, to understand this inhibitor to an inhibitor, right? Because the term, whenever we hear like an inhibitor, we assume that it's a drug molecule. Then what made this inhibitor to block? Why we have another inhibitor? How can a protein can behave like an inhibitor? So there are multiple such questions do arise, but yes, based on our understanding, we can get to know like, yes, we need to develop an inhibitor to an inhibitor. Another protein we took is SWP26. It is also a sporval protein present in the immature and mature spores. We understood something about this. It belongs to, it has a heparin binding motif. then plays a specific role, and also it is there in the SWP26, interacts with the AG-like protein called BMTLP, Bombyx mori turtle-like protein. So this is also a kind of information that we have to use, okay, whenever we design the experiments. They mentioned like this protein SWP26 is already there, and which acts like a target, and this protein is interacting with another protein, that is Bombyx mori turtle-like protein, and this complex which contributes to the infectivity of the Nosema bombyces to its host silkworm. And even I have put the reference paper numbers here also, or if it requires we can also go through. So the reason is, whenever we do with the dry lab, okay, we have to think of two things. One is the quality and also the quantity, because in few cases we will have the docking or anything done, but most of the time people do comment like the quantum of work is less. As I mentioned, it's a project, okay, but it's a research project, but assigned as to one student as a part of the HM project. So don't be in a assumption that dry lab is just click and get the results done, and it won't take, one cannot generate a huge amount of data. It is not the case. As long as we know what are the things that we can cover it up under the study, we can keep increasing both quality and also the quantity. And in that sense, to design those experiments, so one needs to consider if we have any such important information available in the literature. So what we will get to know from this is this protein is interacting with another protein. So this gives us a hint that we can conduct experiment like protein-protein interaction, not just the protein-ligand interaction, so that we will get to know how this SWP is binding. And there is also a paper available to give more confidence in selecting this particular target, which was long back in 2008, okay, and it was clearly mentioned that SWP26 could be used as a good prospective target for diagnostic research and drug design in controlling the silkworm. So these statements from very good journal publications gives us a confidence in selecting the protein as a target. Okay, so based on all these things, we took the SWP26. I'm just showing you the typical procedure that we have followed. I'm not showing you all the results, whatever. Some of the important results that we can look for, and helps us in better understanding or designing of our experiments and how to interpret the results. In that sense, I have put some results. So cellular location where it is present. So SPN106, yes, it is a secreted protein. They showed like, yes, it is there on the outer layer. Then on the cell wall, we have the SWP26. Spore wall protein is there on the SWP26. So you can also get to know from this that we can get through bioinformatics where exactly that particular protein is present. Okay, that is the intention why I have put this. So one can get to know whether the protein is there within the cell or is it there on the cell wall, where it is, or is it there on the extracellular region, where exactly that particular protein is present, that we can. So this is the overall methodology that we followed. We took the sequence retrieval from UniProt, then selected the template, homology modeling we performed, then energy minimization, then model evaluation. So this process itself, okay, took around 1 month time, because usually you might have heard that protein structure prediction is just using Swiss model is a click and go option, and should be fine to get the structure done within a daytime or within a few minutes. But when it comes to a real project, okay, and to cover the entire protein, it takes huge amount of time, sometimes like 15-20 days or a month's time. Or in few cases, we may not get the model at all. We may not be able to get the model that we can use it for the further study. So then we obtained the models of the 3 proteins. Then we performed protein-protein interaction between SWP26 and bomb-exploded turtle-like protein. Then we predicted the binding site, we performed the virtual screening, then we did the lead optimization, then we took it into the molecular dynamic simulation, then we interpreted, we decided which one we should consider it as a lead molecule. This is all with the protein section. So we need ligands for the study. So we took the related ligands that I will discuss. What is the strategy that we applied to get the related ligands? Then we used the Zinc 15 database. Then we selected some ligands from this, 28,500 ligands. Then we made it like a lead library creation, and then we did the virtual screening, nothing but the docking, and then lead optimization, then the MD. So the whole procedure I will go step by step, okay, so that you can also get to know how you can design the experiments and how we can perform and what kind of results we will get. This is what the whole procedure as a overview that we followed. First, we retrieved the sequence from the UniProt SwissProt, and this is SPN 106 having like 384 amino acids. This is the SWP 26 having 223 amino acids. And one protein that I mentioned is the BMTLP. Okay, so BMTLP, bomb-exploded turtle-like protein, is a quite big protein having 447 amino acids. And this protein sequence is not available in the UniProt. Okay, we obtained the sequence from the silkworm genome database. So we may need to use other database sometimes to collect the data. And also, if we observe, not both the proteins, okay, whatever is there, not both the proteins are there under UniProt SwissProt. In the sense, one is there as a unreviewed, okay, and this one is a reviewed data, because some information about this WP26 is available. So it is there under review database SwissProt, but it is there in the Tremble. Not much of information is not available in this. But still we took, though it is having a less. Now, how did we construct the model, homology modeling? So since the PDB structure is not there, we used a Swiss model and modeler. Both we used. First model was generated using online server Swiss model, but it was having a lack of the full length. It did not cover the entire sequence, whatever we wanted. That means we wanted to have a protein structure having 384 amino acids or 224 amino acids or 447 amino acids. But from the Swiss model, we did not get. We got a partial structure. Then we used modular version 9.18. Then we used some templates that we identified in the Swiss model. Those templates we used it in the modeler. Then we performed BLAST-P to identify is there any other templates that we can use to build the model. Then we also used a modeler which has the inbuilt database called PDB95, and then using this also we aim to get the model. Then modeler 19, modeler version 9.18 was used, and then we created this PIR database, and then we have used the Blossom 62 matrix. We have put the gap penalty -500, -50 to get the closely related sequences. Then again the blast search. So it's a tedious process that we followed to get the final model. Download all the mentioned PDBs. Then we took the 5 different models and we selected the one with the least dope score. So if we just look into the whole procedure, right, it is like a bit tedious because there is no single server which will give us the better result or the best structure which covers the entire protein sequence. Swiss modeler, different possible ways. Now we have like iTesser is one server, RaptorX, different intfold. There are servers available where we can submit and get the structures. But we have generated the structures from different servers. Okay, I have just put here the one which is working. So don't think that this is the standard procedure and we will have the results if we follow like this. We need to use all possible servers and based on the kind of results that we obtain from each server, we have to take a decision which is the right way to get the model done. So it's a completely a trial and error method, okay, based on the results that we obtain. Even in few cases where I have done, we got a good result from the ITESR, and if we have a mutant, if I just make one mutation, ITESR failed to give a good result, but for the mutant, intfold is giving a good result, okay, another server. So that means for the wild type, ITESR is giving a good result, but for the mutant, only one amino acid change that happened. ITESR shows a bad result, but intfold is giving a good result. But intfold is failing for the wild type. So one needs to take into consideration different possible ways, all possible servers or the tools available to generate the model. Then we need to check the results of all the servers or the softwares that we use. Then one needs to take a decision which one is appropriately good so that we can continue the study. So this is how the result we obtained, and 5 models that we obtained. And then based on the dope score, the one which is having the least dope score, and the one which is close to the one in the GA341 scores, or the one which is good with the mol PDF score. So the dope score is discrete optimized protein energy. So the lesser the energy, better it is. So we also need to consider those. If you are a new one, okay, let us say if you do not know about these scores or anything, we have sufficient help manuals available, and they do give us the information like which one we should take it. Okay, so it is very clearly mentioned in the manual of the dope from the modeler itself, and clearly said like the one with the least energy is a better structure. And in the GA341 score, they said like if the value is 0, that is the worst or the poor structure. The value is close to 1, that is the good model. So just those information if we take at the first instance should be enough. If it requires, you can go one step ahead and understand how they generated the dope score. But at the first instance, if you are doing it for the first time, okay, just get the information, just understand how to interpret these results. Then you can go to the next level. How did they calculate the dope score, equations or considerations, all those things. So as I mentioned, okay, we have used different models, different servers, and we, based on the Ramachandran plot, we have decided which one should be considered as a good one. So this is just a representation for the SPN 106. Okay, we also made a decision whether to consider it or not. Which one should we consider as a good model? So we looked into the outliers, the one which is having the less residues, that is 6 is there in the outlier, 1HLE from the PDB95 database, 1HLE as a template having less number of residues, only 6, and how many residues it is covering, amino acid coverage. There are few, if we use with the Swiss model as I mentioned, it is covering only 359 amino acids. So SPN 106 is having 384 amino acids. So 359, though the score is good, okay, but query coverage is less. So we said not to go. 338, less than this, no. Then we have a situation 384, all residues are covered, less number on the outliers, and also we checked into the process score and other things. Then based on that, we have decided like this should be the best model that we can take for the drug designing. Same continued with SWP26, and also checked, we generated, looked into everything, which one is good, and then which one we should consider for the next. So based on all these things, there are two situations where it says like 223, it is covering entire protein. Then one can take a decision that which model we can take, and then go ahead again based on the process score. So we have decided that this should be the best one to consider and go for the further study. BMTLP is another protein, which is a big protein, 447 amino acids. Then again, based on the Swiss model or a modeler, okay, different servers looked into. Then we got full protein covered, 447 amino acids, and a good process score, and less number of residues in the outlier in the Ramachandran plot. Then we have decided like, okay, we have found the template, or we have found the structure for this. So it's a tedious process, okay, but as long as we get the good structure, a confident structure, till that time the iteration will keep happening. So the model with the least number of residues is considered to be the accepted, and we also looked into the Z-score. Further energy minimization of the protein was done using mod refiner, which works on a two-step method. This is additional information to know how exactly the mod refiner is going to work. So it first looks at the structural accuracy and then optimizes the side chain. And we used Rampage, Prosa, different server to generate the Ramachandran plot using Rampage and Prosa score. So how exactly the Ramachandran plot looks like? So this I think most of are aware of. So we have like favored region, additionally favored region, allowed region, generously allowed region, disallowed region. So there are two residues in the outlier, and there are 95% in the favored region. So it's a good model. And then we have around 91% in the SWP26, two residues in the outlier, and we have 93% in the BMTLP, which is a big protein, as I mentioned, 417 in the allowed favored region, 22 in the allowed region, 6 in the outlier. In all three cases, we have more than 90% in the favored region. So most of the time, whenever we communicate for the publication, the reviewer looks for this favored region. As long as we have 90% and above residues in the favored region, so even from the reviewer perspective also, they have a confidence that we obtained a good model. Okay, so that also we need to keep in mind. Then we looked into the Z-score. Okay, the quality, model quality we can check. So in the Z-score, this provides us the information how is the modeled protein. If we check this modeled protein with the experimentally determined structure, so the dark blue color spots represents the NMR, and these are the X-ray. So based on the length of the protein, and we were based on the Z-score that is obtained, -4, that is here. So this gives us the information, what if I determine the structure of this SPN106, where it will fall. So it is matching with almost like a NMR structure. Okay, so the theoretical model is matching to the experimentally determined NMR structure score. Same with the SWP26. Here we can observe it's matching almost with the NMR structure. We generated for the BMTLP also, then almost with the NMR structure. So probably even with the further refinement or best template, if we get, then it may go to this region. Okay, so this is the NMR, it is matching. All these are the X-ray structures, so matching almost with the X-ray structure here. The dark one is the NMR. Okay, so it's matching almost with the NMR structure. So X-ray over here, these are all the NMR structures. So even if we look into this, right, typically any one of these, all these background spots is the same, there is no difference. So higher the length, so we have the range of the Z-score also increases. In few cases, we may get a spot somewhere around like this. So we have another situation where the spot coming out like this, because we do not have a template available, but still we have generated the energy of the molecule is good. So then we perform the active site or binding site prediction using CAST-P and oxide scorer. And then from both the servers, whatever the result we obtain from the CAST-P or dark side scorer, we made a consensus. Consensus in the sense, the residues which are common in CAST-P and the dark side scorer, those residues are considered as the binding site residues. So these are the residues for the protein A, that is SPN106 and SWP26. This is also one of the strategy by which you can identify a binding site. If you have like 4 or 5 servers, you can use 4 or 5 different servers and then look at the results and select the consensus, the amino acids which are common in all 4 servers, because we have a higher confidence in those residues. Then we performed protein-protein interaction using PlusPro server. We put SWP26 as a A chain and BMTLP as the B chain. Then we got the results, and again based on the energy we can select one good model. Then we visualize the interaction using PyMol. So here we can get to know like the SWP26 and BMTLP, mummic murine turtle-like protein. How is the interaction? This portion we can zoom in and say like how is the interaction between these two. So we will get to know whatever the interaction that is there here, okay, the region where the interaction is there between BMTLP and SWP6, those residues and these active site predicted or binding site predicted residues, are they overlapping or are they different? So that is what we will get by performing the protein-protein interaction. Is it the same site where this protein also going to bind? That we will get to know. So then we perform the blast P for both the proteins SWP26 and SPN106 to know is there any other protein. This is just to know whatever we did previously is majorly to know structure prediction. Now we want to see which motif is there, which domain is there in the protein. So using blast P when we performed, so we got to know like the protein, okay, belongs to like a serpin family, serine protease inhibitor family, and these are the some of the related proteins. But most of the related proteins, whatever we get from the blast result, we all are predicted are a hypothetical proteins. So again, not sure or not significantly, not having a higher confidence in saying, but there is one which shows like it belongs, same sequence, so 100% identity, but almost all are serine protease inhibitor with the different organisms, and they are not a curated one, they are hypothetical. Then we also checked into the crystallographic, is there any related protein structures available, because we wanted to see what are the serpin-like protein structures that are determined and stored into the PDB. So we performed this. We also conducted this, the result from the blast P, okay, so that we will get to know how is the relatedness, and we also get to know this information. So this has a serpin superfamily. So this protein serine protease inhibitor 106 has a which family it belongs to, so that we will get from this. Then superfamily information we can take. Then this is a crucial step. There are some drugs which are available, or there are some inhibitors which are available for the serine protease. So we took these ligands, okay, these are the PDB IDs, and all these PDB IDs belongs to a serpin family. The reason why we did this is we have like billions of compounds in the database, and it is impossible to take all those billions of compounds and do the screening. So we need to put some strategies to reduce the number and then use it for the virtual screening. So one of the approach that we used here, there are multiple approaches, okay. One of the approach that we used is, because there is no drug available, but still we want to do, so one of the ways is, as this protein belongs to a serpin family, is there any bound ligand, okay, is there any ligand which is bound to these serpin family proteins? When we looked into all the proteins that belongs to a serpin, serine protease inhibitor family, we got some ligands which are crystallographically bound. That's nothing but a co-crystallized ligands. So these co-crystallized ligands information that we took, it gives us the information, thus these ligands are having affinity to bind to these subfamily proteins. We want to know what kind of ligands bind to this protein. So to get that information, we took all the bound ligands from the crystallography. Then we made a list of those ligands. Then we looked into the molecular weight, the range, the number of hydrogen bond donors, acceptors, 6-log-P, heavy atoms, area, everything. Then we also made the range. What is the minimum and maximum molecular weight? Minimum, maximum hydrogen bond donors, acceptors, minimum log P, then the donors like this. And these values are used in this screening. So in the zinc 15 database, we have like 330 million compounds, which we can download for the in the PDBQT format to perform the docking. But considering this 330 million compounds and docking becomes too tedious, so we need to have a strategy. That is what we applied. And then by placing all those constraints, okay, parameters, pH, charge, category, molecular weight, log P value, whatever we observed in the previous. So by giving these constraints, and the number of compounds got reduced to 28,500 ligands. So this is how we can screen in the zinc. We can give all this molecular weight from 200 to 350, the charge we can give, and we can also say like only clean. We need to have a 3D structures. What is the charge that should be? So different criteria that we can give, and based on that, we got something like 28,500. So out of these 28,000 ligands that we obtained, just for the practical aspects to conduct, we split into two batches. Okay, 2,000 and 26,000. We used Autodoc Vina for the virtual screening for the docking, and then we set the grid box where for those bending residues and did the docking. Then when we performed, this is how the grid box setting will be. So we will select all the binding residues that we got from the CASP or doc set scorer. Those residues we can select, label as atom, then we can set the grid box. So what we are doing in the grid box is we are asking the two ligand to go inside this grid box and interact with these amino acids. So it is something like giving a pin code to a letter, okay, where it should reach. So the ligand may be there anywhere on this particular screen when we load, but where it should go and interact, that is what this grid box is going to do. Go inside and interact within these residues. So another thing that I have added here is to just to get the feeling of how much time it takes to do these simulations. So we had like two batches. Typically each ligand, when we use with the Autodoc Vina, it will generate 9 conformations. So for 9 conformation generation, it is going to take approximately 45 seconds for one ligand, one protein interaction. So based on the complexity, it may go to 1 minute also. So if we take into this consideration, it's like 28,500 ligands. So for one protein, the runtime will be this much, keeping into like 45 seconds per ligand. So it is going to take 356 hours. So typically 15 days time for one protein, 28,500 ligands. This is the runtime, okay, assuming that you will sit continuously and perform. If we have 2 proteins and if you have one system, okay, it will take approximately 40 days time to overall to complete the whole docking simulation. Of course, if we have a GPU or other stuffs, okay, parallel computing, if you have 2 systems and one, then parallel you can do and get the results done within 10 or 15 days also. Assuming that if you have one, this is just like if you have one computer, okay, these many ligands, how much time it is going to take. Also another reason why I have put is one should not think that you will get the results within hours of time. The reason why I'm mentioning all these things is there is a huge tendency in the wet lab people that dry lab is just click and get the results, okay. So just to avoid all those assumptions and thinking that why people need weeks time or a month's time to get the result in a dry lab, you should get in a day's time. That is not going to happen. This is how typically the time it takes, okay. Of course, if somebody gives a very good server and other steps, we can take more ligands. and then we can also conduct parallelly. But if we have like a 8 GB RAM and computers, then it takes this much amount of time. Having said, there is no power cut, no interruption, no errors, anything. If those things are there, then we have a more time it is going to take. So from after the docking, we took the 15 top ligands based on the energies minus 9.1 to this is for the SPN106, this is for the SWP26. So the zinc ID is mentioned here, then the Venus score. So the next step what we did is, this is the lead identification. We have identified the ligands based on the energy. Of course, there are many ways. Even in my own cases, we have not just considered the energies, we also look into we have generated the plots for each of these interactions and then selected the right ligand. This is overall. There are different ways by which one will look for. There are few who ask, like, we also need to consider the standard error here, not just the least energy we should take. One should generate the standard deviation for all 9 conformations. Based on that, of course, there are different methods or strategies that one should use, but we just followed at this. In this particular project, we selected the one which is having least energy as the first step in the lead identification, then performed the lead optimization, then we took the top 15 ligands, performed the virtual screening, we looked for the substructure search. So what we did in a overall view, if I just take, so if this is the lead molecule, okay, so we just removed off some of the functional groups and look for the substructure search. So if this is the molecule, okay, so we just removed off this Cl. The rest all we kept. So just we removed some of the groups, this oxygen, so that is there any possibility that we can derive the substructures. So we look for the derivatives, in other words, other substructures that are possible. So when we did like this using PubChem, we found like 633 additional compounds for the SPN106, 859 for the SWP26. Then this is like a lead optimization. Can we derive new compounds having the same moiety or the scaffold? Then we performed the docking of all those ligands. Then we found that these are the ligands having the least score. So it moved, if you observe now, it moved from minus 9.1 or minus 8.7 to minus 12 or minus 10. So there is a further development, improvement in the affinity between the protein and the ligand. Then these top identified lead molecules, we took it into the dynamic simulation. This is the structure that we identified. Then that we took it into we looked into the leaf plot to see how is the interaction between the protein and ligand. So we generated the leaf plot. We observed that the ligand forms 2 hydrogen bonds, one with the lysine 31, one with the tyrosine 245. Same with the SWP26. Okay, there are 1, 2, 3, 4 hydrogen bonds, glycine 64, tyrosine 77, 72, split hydrogen bonds, then the 85 position. So another thing what we observed, or whenever you are doing, okay, whenever we have the results, one should look, is there any ligand which is common? So it is a coincidence, and in the coincidence case, it is observed that 129028069, the same ligand is there here. That means we have a common ligand which can bind to SPN106, okay, and it can also bind to SWP26. We also observed another ligand 11202059, okay, and also the same 11202059. Other ligands are not. So what it says is we can use one drug molecule which can bind to both the targets, both the protein targets. So if it fails in one, it can give a good result in another one. It is like a single drug molecule binding to 2 proteins. That's a good result in the perspective of the drug designing. Then we performed the dynamic simulation using Desmond. Desmond is free for the academic purpose. Then we looked into RMSD, RMSF, radius of gyration, residual interactions. We did the dynamics for 100 nanoseconds. Then we looked into the RMSD. How is the stability of the protein? Okay. So root mean square deviation, this tells us the one which is black color is the APO. APO refers to without ligand. So it is having a slightly higher deviation, and the deviation of the protein is decreased when the ligand is bound to this. That is red color is the. So the deviation, deviation of the protein is reduced when the ligand is bound to this. Now, which amino acid actually undergoing more deviation or supporting this deviation to happen? That we will get from the RMSF. Okay, just a second. Okay, so that we will get from the RMSF. I think the RMSF of this is not okay. I will just check. So RMSF provides the root mean square fluctuation, where exactly the deviation is. Then radius of gyration. Radius of gyration tells us how is the compactness of the protein. So if the value increases, okay, so if the value increases, it is losing its compactness. So this has more complex, that is the compactness is good here when it is APO. When the ligand is bound to it, the compactness of the protein is reduced. Then we can also see how is the interaction between the ligand and the protein through the histogram. So the ligand is having a strong hydrogen bond with the aspartic acid 92, and there is some hydrogen bonding with the tyrosine 89, then GLN 85. So that also we will get to know where is the interaction, whether it is hydrogen bonding, whether hydrophobic interaction, is it ionic or is it a water bridge, all that we will get to know from the Desmond. Then we also performed, this is with SWP26, okay, this is with SPN 106. So we have generated the plots for both SWP26 and SPN 106. Then based on all these things, we made a conclusion that this ligand was identified as a lead molecule, and then one can use it as a drug-like molecule. And there are some significant hydrogen bonds. There is a increased stability of this ligand with both SPN106 and SWP26. Okay, so based on the docking results, the dynamic results, so we finally said like we can use it as a drug-like molecule. And now we are into a stage where we are testing. We have the silkworms. Now we will procure this particular compound, and then we will infect, then take these proteins of our interest and then test whether it is going to inhibit or not. So there are multiple proteins available. Okay, so we took other proteins also like SWP25, and this SWP9 can also be used as another target protein. Nosema bombaceus in our RBL can also be used as another target.
ऑक्स का ऑफ मैंने बोला ऑक्सिस तो कह रही हाँ ऑक्सिस होता है ध्यान से ऑक्सीजन जो मैं सोच रही थी.
Okay, so these are some of the references that I have placed over there for easy understanding. And this particular work, whole work, we published in the Taylor and Francis Journal of Biomolecular Structure and Dynamics journal, having 3.3 impact factor in 2019, last year, and came into published in the month of December. Okay, of course there are some amount of review that happened, a lot of revisions happened, and some sentences, some experiments also we were supposed to do. We initially did for 50 nanoseconds, then they said like 50 nanoseconds is not sufficient, we need to have it for 100 nanoseconds. So we reconducted, we reperformed with 100 nanoseconds. In few journals when we conducted, they do ask like better to conduct triplicates. Okay, so we need to conduct sometimes in triplicates for 100 nanoseconds and then decide the so it goes. Okay, so we do not know the kind of a questions we will get from the reviewer. But whatever the facility available, whatever that we can do, the best way of interpretation based on that. But yes, this is just a work that I'm showing here to give you an hint that even in the academic section, without using any of the commercial tools, completely with the open source, one can aim to publish in the high impact factor or Scopus indexed journals. Okay, thank you.
Hello.
Yeah,
Sir,
so
thank you, sir, for your presentation.
yes, yes.
Valuable information and guidance for the participants, I hope. Sir, questions are posted in the chat, sir. You can go through it one by one.
Okay. Okay, so let me look into one is from Dr. Okay, Praveen Kumar, is crystal structure available for SPN 106? No, there is no crystal structure available. It's completely with the homology modeling method that we obtained the structure. In the next, that is what I am mentioning, that is in 2018 work. In 2019, okay, I took another student where we could not use even the homology modeling. Okay, there is no template available that is with SWP5, another protein. So we used threading method to get the protein structure, and that was much more tedious process. Ab initio or threading method, almost like a ITSR, and then further refinement with the mod refiner, galaxy refiner. Then we obtained a reasonably good model. Okay, if you want, I can show you some of the results. And just like BMTLP, okay, which is having like 446 amino acids, we have two other proteins, PTP2 and PTP3. And PTP3 is having 1336 amino acids. So we built the model for 1336 amino acids using ITSR. And it is accepted, okay, in the sense it's almost accepted. So though it was, but we can aim, okay. There are some review comments, of course, we will get, because people think from the perspective of the reviewers, they do comment that the number of residues in the favored region in all three proteins that we used recently, okay, is 86%, 87%. No protein crossed 90%, but it is an attempt. All possible servers, algorithms available we used to get the good model, and it works fine, and it is going to be accepted. Okay, first reviewer is accepted, should come within a one step. But we need to justify. There is no group in the world working in the protein structure determination, and we cannot wait for someone to determine the structure for this and then for me to use it as a template. So we do not know when it is going to happen. We have algorithms, we have tools available. We may not get as accurate as that, but if we get the results, if we are getting a prominent result, see, we cannot always just go with the Ramachandran plot. We have verified 3D, okay, PROSA score. There are different ways by which we can validate whether the structure obtained is correct or not. So we will use all possible methods that are available for the validation of the structure. So if we get a good result, yes, we can claim that the model structure is correct. Okay, so then the next question, there might be some novice like me in this field for some introductory discussion on the basics of this high-end topic would have helped to understand it a bit. Correct. So that is why I just initially mentioned with the molecular docking, though for few it may feel like too technical or too much in detail, or maybe difficult to understand at this point of time. The reason why I have put the completely technical aspect is it is not required for anyone to know the entire stuff at the algorithmic level. If we get to know that these are the possibilities, these are kind of a possible collaborations that one can make, or this is quite possible that one can do and still publish. So that itself is a more than enough. Just getting a hint that yes, just with homology modeling, docking and dynamic simulation, yes, it is possible to publish in a journal having 3 impact factor. If you can get that feeling, my job is done. Okay, so my purpose of whole presentation is that only. You can aim for a good journal, okay, even Springer or Elsevier. Of course, in few journals they do ask with the wet lab validation. If we can do with the wet lab, it is well and good. If it is not possible, still there are some good journals, okay, which undergoes a thorough review. Okay, so in both the cases for this or the recently communicated, I have received around 53 comments. Okay, each and every line there are some comments, but it improves the overall paper. And sometimes we really need to conduct the experiments, redo the experiments. It is fine, we can conduct again so that we have a higher confidence in the results. So you don't need to understand. Okay, so as there is one of the things from Dr. Shukanti Bhattacharya, that it is difficult, some introductory discussion, as we have only 1.5 hours time. The main aim, as I mentioned, is just to understand the possible ways by which we can design the experiments and possible ways by which how we can write and publish. And also I need to mention that the work is not completely by, I have guided, the student has performed. Okay, so one can think of giving these as a 8th sem projects, and you can just need to train them in few aspects, they will perform, and we can aim to publish the project work in a high impact factor journals. How can we practically use this drug? How we can test this using wet lab experiments? Correct. So we are doing this. In fact, we also got a funding, that is from the internal funding, to test this with the wet lab. One of the ways when I discussed with the collaborator is to check the typical melanization pathway. Okay, as I mentioned here, and okay, so there are ways by which one can do the test this melanization pathway. Okay, so if this melanization pathway, if it is normal, how is the rate? And when we infect with the serine protease inhibitor, how much it is going to affect, that we can test. So this is quite possible to conduct in the wet lab. Then the drug that we have designed against this, that also we can check whether it is going to inhibit or not. So we can, this is not difficult, this is feasible to conduct. So tools for molecular dynamic simulation studies, we have used Desmond, okay, from the Schrodinger, and for academic purpose, Desmond is free. Okay, one can download from the DE Shah website. You just need to register from your academic mail ID, and then you can download, and one can work. So it is quite possible. So my mail ID, yes, it is already, I have put that one in the first slide. Okay, so this is my mail ID. Sorry, prashantakarunakar@pes.edu. Okay, so this is my mail ID. Sorry, and this is my contact number, 81055478886, and this is my personal website. Okay, sets.google.com. So even there is an option in this site where you can, I have created a form where you can give your phone number, mobile ID, and the kind of a problem if you are facing either in docking or dynamic simulations. You can post that one, then I will receive the mail and I will contact you or reply back to that one, giving the best possible solution that I can do. Then sir, in your presentation, you showed 7 ligands bound to SNPs. Did you use those or from Zinc database library? Bound to SNP or SPNs, did you use those? Or see, the 7 ligands are which are bound to the serpin family, serine protease inhibitor family. So those ligands we have considered as a library or initial set to get the range of molecular weight, log P, or the charge. So using that as a parameter, we screened the zinc. So these are the crystallographic one. Those 7 are the crystallographic. They are already bound to the serpine family. So that we used. It is something like if I want to design a new drug against the coronavirus, or if not with the coronavirus, okay, because there is no drug. So if there is anything with the cyclooxygenase or anything, if there are drug molecules in the market, we just need to understand initially how exactly the drug molecules, physicochemical properties are there. So once we understand its physicochemical properties and its corresponding side effects, so it helps us to decide active and inactive basically. So which compounds are safe and what is the corresponding side effect. So we will get to know the difference in the structures and corresponding levels of side effects. The one which is having a serious side effects, how is the functional group, how is the pharmacophore. Then we will get to know, like when I design a new molecule, I should not keep these pharmacophores because it will, again, I will also have the same side effects. So that is one of the ways. In that sense, we took the drugs or the ligands which are already bound. What we are doing, people can use the molecular mimicry concept. If ATP is going to bind to the protein as a substrate, we can use the ATP as a reference and then derive the mimicry, okay, molecular mimicry. Then you can use it as a drug molecules. So different ways. What can be methodology if we are screening medicinal properties of plants and we have plant metabolites identified through GCMS in hand? Yes, we have I have a paper on this one for each of these things. When you have a GCMS done, okay, you have identified the ligands, and if we have a wet lab data, that means if we have done any of the biological assays, so you know how. Let us say in our case, okay, one of the paper that we have published. So the kind of a work is initially my collaborator performed the plant extracts. I think they took the seeds and the shoot and stem like this, and then they checked whether they have any medicinal activity, biological activity. Then it was sure that it is having. Then the next step is they did with the GCMS and then tested in vitro. Then also they got the results. Then it is with anti-cancerous property. Then we took all the GCMS compounds, which were around 48 ligands that they have identified from the GCMS. Then out of 48, just check for how many compounds in the literature we have a proof that they are having an anti-cancerous property. Okay, then we took those compounds and then select the appropriate target protein, then do the docking, then say which one is really acting. Yes, it is possible. We can, from the GCMS, we can perform the docking and then go for the next dynamic simulation. One thing I should mention here is in most of the cases, okay, if we go for a good journal, okay, like a Taylor and Francis or any of the journals, dynamic simulation is a must. Okay, it is becoming like a mandatory step now. Previously, the docking was sufficient to publish. Now the dynamic simulation is coming out, and even in the recent publication or recent communication when I made, people are also asking with the heat map, which people never asked before. Heat map is like how is one ligand behaving, okay, over a period of time. So if I check with, just to give you, okay, so the expectation is increasing. That's what I'm mentioning overall. So if we have this RMSD, so the deviation is happening. Let us say the deviation is happening over here or deviation is happening here. So the expectation is something like which amino acid is making this fluctuation, that we will get from the RMSF. But how is the behavior of that particular amino acid throughout the 100 nanoseconds of time? So that also we need to do. So the expectation is increasing. That's what we need to look into. Binding energy, MMPBSA, MMGBSA, we need to do a lot, and the expectation is increasing. So can you suggest few more open source tools for modeling? Most of the homology modeling tools are uh, open source. We use the ITSR, Inkfold for, and then RaptorX, Rosetta. Okay, in most of the cases, I found ITSR is working good in getting the model, and in case of modeling, docking, docking is working fine. Autodock is working fine. Okay, we are getting a good results. And for the dynamic simulation, Gromax or the Desmond, both are good methods. Gromax is a good tool, open source, free to use, and yes, but in few cases we will get some errors to troubleshoot, and we do not have a GUI. One more thing we also need to is we are a kind of a biologist. We are not like a computer science background, so we prefer mostly with the graphical user interfaces. So and as long as we get a good result, so it's fine. GUI, if it is there, we can visualize there itself. So Desmond has that feature, so we go with that. But Gromax, yes, over a period of time, though it is a command line, it works really well, and again goes with the kind. Most of the time when we communicate, if we get some comments like generate the MMPBSA and other stuff, then also we will perform with the Gromax. This was from Dr. Swati Madhe. The next question is from Dimfan. Can you mention about the choices to be made when a mutant protein gives different results for different tools or parameters? Of course, when there is a when we have a mutant protein, I recommend you to fix with the tool. Okay, based on that only we can make some inference. For example, using docking with the gold for native and Autodock for the mutant, we may not be able to correlate. So we should not change. Okay, so the tools should be as consistent as possible so that we can make some inferences. But when we are selecting the results, like model selection, use all possible servers, take the best one. For mutant proteins, yes, choices to be made. Can we mention about the choices to be made when a mutant protein gives different results for different tools or parameters? And slightly, the results will be different, okay, because we have a mutant proteins. This is fine. And when you use a different tool, again the different results we will have. When you change the parameter, again we will have different results. So there's no surprise in that. But what I am saying is keep the tool constant. Okay, Desmond, Dynamics, Autodock as a docking. If you fix, keep all the parameters same for the wild type and the mutant. Don't change. And then the results, whatever you get, there will be a difference that we need to interpret. So it is not good to change the tools or the parameters when you are working with the native and wild type. Keep everything same. There will be a difference in the result using the same parameters or the same tool because there is a mutant protein. So its effect that we need to understand. So it is the same like whenever we work with the multi-drug resistant proteins. We take the wild type, we take the drug resistant protein, then look for where is the difference. How is the drug binding to the native protein? How is it binding to the mutant protein? Where is the difference? Why it is not acting? What made it to have the resistance? So we can make all these interpretation only when we have the same parameters, same tools used. Otherwise, it is too difficult to make the interpretation. Another question from the Sanjoy. Is there any software for running molecular dynamic simulation for Windows 7? There are, okay, but even Desmond, you can use it under the Windows 7, but it is preferred to run in the Gromax, the Linux system, and because we have multiple options where we can write the small commands and get the results done. And yeah, the efficiency is also very high with in the Ubuntu system or the Linux system when compared to Windows. And there are many bioinformatics or docking or dynamic simulations supporting for the Linux than compared to Windows 7. So better to use the Ubuntu because there is a dependency from one to another tool, because output of one tool is the input for another tool. So there is a huge amount of dependency, and most of the tools supports for the Linux. So it is better to use the Linux just to finish the tasks or just to have the experiments done in a quicker time. Otherwise, we need to switch from Windows to Linux, again Linux to Windows. So it becomes a bit tedious, and file transfer, file saving also becomes difficult. But GROMACS needs a long time to run protein for hundreds. Yes, of course, it takes long time. 100 nanoseconds, it is going to take long amount of time. So that is why we may need to collaborate with the one who is having the servers, like NVIDIA graphics card, if they have. If we do not have the facility, the only way to get the results done is to collaborate or pay and use option, okay, as a consultancy. So we may need to go either of the ways. In few cases, the collaboration people may not ready, okay. So in such situation, we just need to go like, please give us the results, we will make the interpretation, we will pay this much amount, okay, whatever they charge. So there are some consultancy agencies for the dynamic simulation. Okay, we can contact them. We can say like we need 400 nanoseconds. These many simulations we need. If it requires triplicates, accordingly they will charge. Or so it goes. If they are ready to go undergo the publication as a authorship basis, then that is also good. If that is not possible, but we want the result at the end. Okay, so we need to find the ways, collaboration or pay and use option. That is another model that we can follow. But if we want to run ourselves within this 8 GB or 16 GB RAM computers for 100 nanoseconds, we need to wait for months. So we may not have that much amount of time from the reviewer. If I do for 50 nanoseconds, usually they may give like 1 month or 2 months time to reply back to their comments from the journal. So we cannot spend 2 months only in the simulation. So the best, and we may not be having an option to add an author when we have communicated, because we cannot add another author. So at that time, only option is pay and use option. Okay, we just need to pay the one who can perform this dynamic simulation and then get the results done. If they are ready to support in interpretation, that is well and good. Otherwise, we just need to learn and then write the interpretation. So that's another option. Yeah. Is there any doubts or anything? I'll be more than happy to address.
Thank you, sir, Prashant, sir.
Yeah.
For clarifying the doubts of the participants. On behalf of the University Benedict College of Engineering from MCA department and E&I department, I thank you once again for sparing your valuable time and knowledge to the participants in this ATAL national level FDP.
Okay. Yeah, it's pleasure. Thank you very much. Thank you for the invitation, and also thanks from all the participants. I hope it was useful. There are quite interesting questions and very interactive session. Yeah, thank you very much.
Yeah, please share your slides, sir, so that we can give it to the participants.
Okay, so this slides I can share, and also the full length paper is also available. Okay, so I have used the same thing. So the full paper itself is available. Like I will share the paper also, that one can use it whenever you write the papers.
Yes. Thank you, sir. Thank you.
Thank you very much. Thank you, all the participants. Thank you very much.